hello, and welcome to today's lecture topic. It's titled Epigenetics, the Microbiome, and the Importance of Regenerative Agriculture for Human Health. This is a very broad topic. Um, so, each one of these entities could encompass an hour-long lecture in and of themselves, but I'm going to try to keep this to an hour-long or an hour's worth of content broken down into 3 parts, and we'll try to stay at 30,000-foot view, and occasionally getting more specific just to prove some points. All right, so let's get started. So the Human Genome Project concluded in 2003, and they thought they were going to find 100,000 genes. They found somewhere between 20 to 25,000 genes, which is similar to other mammals, but less than flowering plants, including most grasses. They also found that about 99% of our DNA is non-coding. And so this is just a graphical representation showing where we fit with the number of genes we have versus other creatures and other species. And you can see that, the flowering plants have a tremendous number of genes. We have fewer genes than a grape. So about 10% of the cells in our bodies are human. That's representing about 20,000 genes. But we have 100 to 400 trillion microbes that are in or on our bodies, and they encompass about 2 million genes. So we are considered a holobiont, an ecosystem that's an assemblage of a host and many other species that live on, in, or around us, which together form an ecological unit and exist in a symbiotic relationship. And so we co-evolved with this microbiome, and most of our historical microbiome colonizers were sourced or derived from the soil and food. And our microbiome assists our body with proper immune and neurologic function and nutrient provision. So it's in the best interest of the microbiome if the host is alive and thriving. So the microbiome promotes the maturation of secondary lymphoid organs. This starts at birth. The immune response, both the adaptive and the innate immune response, require input from the microbiome. And so, the immune system needs to be able to distinguish between our commensal organisms versus pathogens. And commensal bacteria can alert the immune system of intestinal viral infection and help prevent local viral replication. And so these are just some studies that demonstrate that the gut microbiome, instructs a poised basal state of our dendritic cells, and actually calibrates the activation threshold of our innate antiviral immunity. And over here on the right-hand side, you can see where dendritic cells in the colon are sampling the contents of the lumen and getting feedback from the commensal bacteria, and are attempting to determine what is commensal, okay, versus what is a pathogen. And so this plays into the concept of oral tolerance, which is the process of continuously exposing our immune system to innocuous outside antigens. So loss of oral tolerance from sterile soils, foods, or environments can result in the immune system attacking food and environmental particles. And the mucosa is the largest surface area in our body, and the largest site of immune sampling is in the GI tract. So the majority of this is occurring in the GI tract. And so, it's very important that we are exposed to antigens throughout our life, particularly at a young age. So this is a study that demonstrated that mice that were reared in germ-free conditions failed to develop immune tolerance and were more prone to allergic type immune responses. And the reconstitution of their intestinal flora with a single bacterium, we have many in our gut, but just giving them a single bacterium restored the susceptibility to oral tolerance induction. But it was only effective when it was performed in neonates and not in mice at an older age. So it's very critical that we're colonized with the appropriate bacteria and exposed to antigens, both pathogens and non-antigenic or non-pathogenic antigens at a very early age. And so our commensal bacteria in our gut also drive the interferon steady state in the lungs. And so, they can increase or decrease the amount of interferon that is produced in our lungs based upon the signals that they are sending from our gut. And so they communicate with the lung microbiota and assist each other with resistance to pathogens. And some of the strains of bacteria in our gut even produce antimicrobial compounds themselves and can act as antibiotics. So the gut microbiota also communicates with the brain to maintain homeostasis and results in the production of short-chain fatty acids from dietary fiber and influences serotonin and GABA levels in our bodies. And so the complex carbohydrates that we ingest are converted by our gut microbiota into these short-chain fatty acids, which are very important for us. So disruptions to the microbiome from things like antibiotics lead to dysbiosis and a decreased immune response. And this is due to both a decrease in numbers and species diversity. Dysbiosis is also associated with a chronic inflammatory state and numerous chronic diseases. And so these are just some studies demonstrating that antibiotics, can have a detrimental effect on our immune response because they're changing the flora within our gut. And this is a study that demonstrated that antibiotic use can actually decrease the efficacy of certain cancer drugs that are being used now that utilize our own immune systems to kill cancer cells, and that when patients that are taking these immune checkpoint inhibitors are given antibiotics, that the efficacy decreases. So, many genes are conserved between species. You can see that, we're 90% genetically similar to cats, 94% genetically similar to dogs, and 8% of our human DNA is viral in origin. So this begs the question, genetically speaking, what makes us human? And so this is the epigenome. So epigenetics is the expression of genes in relation to the environment in which the individual resides. This starts before conception and extends to death. The epigenome is heritable in non-Mendelian fashion and can be transgenerational, and genes can be expressed anywhere from 0 to 100%. Rarely are they expressed at a 0% or 100% level in any given cell. And so some examples of epigenetics are the cells in our body. So every cell in our body has the exact same DNA, but has a different phenotype, looks different. So, our heart cells look and perform very different from the cells in our eyes. Prader-Willi and Angelman syndrome are two syndromes with the exact same deletion at 15q on the short arm of chromosome 15q, or the short arm of chromosome 15, but have dramatically different phenotypes depending upon whether the chromosome, the deletion, was inherited from the mom or from the dad. And so Prader-Willi, children with Prader-Willi syndrome are overweight and have insatiable appetites. Children with Angelman syndrome have a unique gait, a unique walk, and typically have a face that looks like they're smiling. And so very different phenotype, but the exact same genetic deletion. And then trisomy 21, which is Down syndrome, has variable penetrance. There are children that are highly functioning with Down syndrome, and there are children that are severely impaired. And so these are just examples of epigenetics, different differential expression of genes. And so just to review, genes are transcribed by RNA polymerase from our DNA into mRNA, and then are converted through translation by ribosomes into proteins. And so epigenetic changes affect which genes get transcribed and their levels of expression. This can be done by DNA methylation. This is where a methyl group gets put on the promoter region of a gene and RNA polymerase cannot bind. Histone modification, where genes can get wrapped up around histones and are inaccessible by RNA polymerase, or inaccessible to RNA polymerase. And by non-coding RNA, such as microRNAs, which are very small snippets of RNA that can bind to DNA and prevent the ribosome from coming across and translating that gene or that mRNA into protein. And so the environment in which we live and our behaviors, such as diet and exercise, can result in epigenetic changes. And this is a dynamic process, it's not static. So this is a study where they compared sets of monozygous twins. They have the exact same genotype, the same genes, and they compared the level of gene expression, the similarity of gene expression at different ages. And so yellow is similar gene expression. So you can see in the 3-year-old twins that they're essentially expressing the same genes and at the same level. But by 50 years old, monozygous twins, red and green is different gene expression. They're expressing different genes, or expressing their genes differently due to the environment and the way that they live their lives. And so this epigenetic DNA imprinting activity is most active from conception to the 3rd birthday. So the first 3 years of a child's life, and which is referred to as the 1,000 days period. And this affects both the child and their developing germ cells. So 2 generations are affected by epigenetic DNA imprinting simultaneously. So at the same time that a child is experiencing epigenetic DNA imprinting in the womb as to which genes are going to be expressed and at what level of expression, their germ cells, their developing eggs and sperm are also undergoing epigenetic DNA imprinting. And you can see there that it's very active prior to 7 weeks after conception. And during this time, the first 1,000 days period, after a child's born up until the 3rd birthday, there's also a high turnover of the types of colonizing bacteria that occur in the gut, and after which the microbiome becomes more stable. And so this is just a demonstration of bacterial microbiome diversity in humans, and that you can see a steady rise and increase to about age 5, maybe peaking at around age 12, and then it starts to decline after about age 70. And again, just to demonstrate the importance of this first 1,000 days, there's numerous studies that demonstrate how important these epigenetic marks are in developing children and in children shortly after birth. Oocyte vitrification, which is freezing, cryopreservation of eggs, has negative epigenetic influences. In vitro fertilization can have negative epigenetic influences. Early diet and nutrition can have negative epigenetic influences on a child. And so this is just a graphical representation showing how nutrition and the microbiome and the epigenome are related and how important they are, stressing again within the first 1,000 days of a child's life. So, microbial diversity is obviously very important, and the influence of our diet and reduced microbiome diversity, which is essentially what we're experiencing now in the Western world, forms the foundation of scientific thinking as to how the allergy epidemic occurred. As we're exposed to fewer antigens, less microbial diversity in our gut, and fewer childhood diseases, the risk of allergy and autoimmune disease increases, and this is most important during early postnatal development. And so to further stress this and demonstrate this, this study took 640 infants that were at high risk for peanut allergy, and between 4 to 11 months of age, randomized them into a peanut consumption or avoidance groups. And the children that were in the consumption group that were being fed peanut products had an 86% reduction in peanut allergy by age 5. Dramatic decrease in their risk of peanut allergy. There's also, this is a study that demonstrates the early exposure of probiotics, which are bacteria essentially, to type 1 and its effect on type 1 diabetes. So 7,000 children that were at high risk for developing type 1 diabetes, they had the DR3/4 genotype were exposed. They were randomized and were exposed. Some of them were exposed to early probiotics. And what they found was a 60% decrease in the risk of type 1 diabetes among the children that were exposed. And this decrease in risk was most dramatic the earlier they were exposed to probiotics. This is a study demonstrating the importance of diet for epigenetics and epigenetic imprinting. So pregnant mice had an altered agouti gene, which made them prone to being yellow, obese, and have diabetes and cancer. And they were fed a diet rich in methyl donor groups, folic acid, B12, choline, and betaine. And what happened was that they were able to, through this diet with these methyl donors, were able to induce methylation of the variant agouti gene. And so rather than the mice, the pregnant mice, producing 3/4 offspring that were yellow and obese and prone to disease, methylation of that gene turned that gene off, and 3/4 of the offspring were brown and lean and not prone to disease. So just a demonstration of how important diet is for epigenetic DNA imprinting. The gut microbiome also changes in response to diet. So for instance, obese individuals cultivate bacteria that enable more calories to be extracted from energy-rich diets. And with time, this may not be reversible. So if the gut microbiome lacks the diversity to produce short-chain fatty acids like butyrate, then prebiotics, which is fiber essentially, our diet, will not be sufficient, and probiotics will be necessary, meaning recolonization of our gut microbiota from food or from stool with the beneficial bacteria. And it's also been shown that obesity may be transferred to germ-free mice by colonizing them with intestinal microbes from obese humans. And so an individual inherits their microbiome from their mother at birth, and therefore the microbiome may become transgenerational. So this is a study where they did a heat map demonstration of gut microbial diversity in hunter-gatherer groups versus patients from the United States. And we had a dramatically decreased level of gut microbial diversity. And what they did was they took mice, colonized them with a diverse array of microbiota in their gut, like similar to a hunter-gatherer, and then they fed them a Western diet, and then they bred them. And after the pups were born, then they fed the parents. They took them off of the Western diet. But what occurred was that after several generations, the gut microbiota, the diversity decreased, and it didn't matter what the diet was. They had lost that diversity and only had a few organisms present in their microbiome. So it was not reversible by diet alone. And it's also been demonstrated that transfer of intestinal microbiota from lean donors, from lean individuals, can increase insulin sensitivity in individuals with metabolic syndrome. So gut microbiome plays a huge role in chronic diseases, and in this instance, insulin resistance, which is type 2 diabetes. So a high fiber diet favors butyrate producing bacteria, and normal colonocytes derive 60 to 70% of their energy from butyrate. Colon cancer cells undergo what's called the Warburg effect and switch to glucose utilization and aerobic glycolysis. And patients with low fiber diets or lack of microbiome diversity may induce this epigenetic switch as well. This is a study where pregnant rats were exposed to glyphosate, that's Roundup, at what the EPA considers half the no observable adverse effect level. They were exposed during days 8 to 14 of gestation. And this is the period of high levels of germ cell epigenetic programming. And so what they found was that the offspring really were largely unaffected, but by the third or the F3 generation, they had significant pathologies, prostate disease, obesity, birth abnormalities. And so like the microbiome, this demonstrates that epigenetic influences are heritable and transgenerational as well.